Everybody today to our webinar. This is session 4 of the NIDDK Data Management and Sharing webinar series. And today we're going to be talking about data reuse. So the R in FAIR stands for reuse, and that's an ongoing topic as we continue to make data, more and more available with the data management and sharing policy. So for those of you who are interested in our previous webinars, I'm dropping a link in the chat to the NIDDK Data Management and Sharing website, where there will be links and information from our previous webinars. So you can go view the recordings and the materials. As Cameron said, the material from the webinar today is going to be available at a later date, and I'll be sending out an email with links to that material following the webinar as it becomes available. If you have any questions, please do enter those in the Q&A box, and we're going to be doing our best to get to those during the webinar and in the discussion session. So can we go to the next slide? Thank you. We have a great group of panelists today that are going to be sharing some helpful information and some resources on the topic. First is going to be Dr. Vivian Odawong from the Office of Data Science, and she is going to be giving an overview of secondary data use and how the sharing of data can really help in our advancing our knowledge. After that, we're going to have a presentation from Dr. Harold Lehman, who's going to give an overview of strategies to find data and some considerations for the data reuse. And then finally, we have Anna von Gulick, who's going to give examples of tools and resources, highlighting those from Figshare to help researchers find data and then sharing that data. So with that, I'm going to pass over to Dr. Odawong. Can we go to the next slide and start getting her deck up? Great. So Dr. Odawong is from the Office of Data Science and Strategy at NIH, where she really works to promote awareness of the ethical and social issues in an increasingly multidisciplinary field and a data-driven area of science. So thank you very much.
Great. Thank you very much for the generous introduction. I'm going to start the session off today really trying to really show you the excitement that is going on related to secondary data use. Next slide, please. And I'm going to do it through 4 separate lenses. The first one is really the drivers of why we're even talking about this. And I'm going to talk about it through issues related to human rights and open science. Next, I'm going to move into what are we actually talking about in terms of data. And so looking at the volumes and the varieties of data and the issue and the broader ways of data and data science. And then the next piece is really looking at challenges that we may actually be either anticipating or currently experiencing on the economic, ethical, legal, and social implications. And the next steps, which is really you. Next slide, please. So let's go ahead and start with the drivers. Next slide, please. Fundamentally, what we're talking about is we're trying to understand what is causing diseases. So this is from the Centers of Disease Control and Prevention, and really looking at the top 10 leading causes of death. And as you can see, the heart disease is really ranked as #1, with cancer in a close second. And as you can see, with the advent of COVID and then the remaining 4 through 10, some are really like accidents and stroke, following up with chronic respiratory disease, Alzheimer's disease, and rounding it out with renal failure. The next slide, please, really shows that a lot of these diseases, there's a people factor to it. And so what we also need to consider as we start looking at data reuse are really the dynamics of modifiable behaviors that can contribute to mortality or not. So the messages are basically don't smoke tobacco, eat healthy, be physically active, don't abuse alcohol, wash your hands, don't play with poisons, don't play with guns, have safe sex, wear your seatbelt, and don't abuse substances. So what does this all mean for the next slide? Is that within this new, next slide, please, is this whole, sorry, if you could go back one, is really the landscape we're talking about today is really the new landscape of science and what we're calling big data. And what big data is really meaning is not only the types of data, but the amounts of data. Next slide, please. So part of this movement, I think we need to keep in mind, is this whole what we're calling democratization of knowledge. And the way I like to think about it is really framed within the human rights perspective, is that everyone has the right to information and knowledge. So as you can see from the schematic above, if you start from the orange on the left, it's really talking about benefits to individual researchers, which many of you may be on the webinar today, saying, how can what I am researching benefit the overall society and our knowledge base? And then going to benefits for the research communities. And those are our colleagues, whether they're in our specific disciplines or across the multidisciplinary divides. And then on the far right, that everything that we're talking about really should benefit all members of society. And we need to figure out ways of doing that. So the next slide, please. So in 2018, there was a study commissioned at the National Academies that talked about what we need to do is figure out how can we make science more open. And so the report, Open Science by Design, was really a roadmap of the ways we can think about this. And so one of the hallmarks was really about data sharing and innovation, with the argument that open access to research data which would include assembling the research and the data and making it available to all members of society should be our fundamental goal. And that not only includes the data that we're actually generating as researchers, but the sources and the algorithms that we're using to analyze the data. Next slide, please. And by doing that, what it will do, it will facilitate innovation of the research tools and methods that we currently use to the next generation. Next slide, please. It will increase the statistical power of our studies to actually give them greater validity. And next slide, please. It will improve our overall research quality through its validation and replication of using the data. Next slide. So how you have been hearing all of this is what I would posit is the whole dialogue and discussions about precision health. And what precision health means is it's really accounting not only for the biological factors related to health and disease, but it's also accounting for the social and cultural complexities that are influencing that underlying biology that our research participants truly live. So what this will require is a biological understanding of phenomenon, that we need to be sure to include social, cultural, and psychological factors into our larger equation. And by doing that, it's going to make several advancements in scientific methods, in instrumentation, in technology. And as we're talking with the theme of this webinar, data management and computational advances. And this will include a lot of different types of data, including omic, which could be genomic, metabolomic, imaging data, clinical data, laboratory data. And what we are starting to see, even in the literature now and in the clinics, with this sort of new wave of thinking about our data, it's starting to change disease classifications and treatments. Next slide, please. So ultimately, what the open science movement and data sharing is doing is at a fundamental level, it's increasing the scientific value of data by having the ability of researchers to explore, to combine, to analyze data from a lot of different sources. Next slide, please. It's also going to increase the scale of studies in terms of publications, the types of scientists from a broader range of disciplines. So as you can see from this schematic, this is actually a demonstration of the impact of sharing brain imaging data. The top bar graph shows in the unfilled bars the actual publications that the investigators who generated the data actually produced. But the filled-in bars are the production of publications of researchers who were doing secondary data analysis. And what the bottom bar shows is part of that secondary data analysis was really capitalizing on a lot of different fields. And so the work is truly multidisciplinary. So you can see that it's spanning from not only from engineering, but neuroscience, all the way to education and medicine. Okay, so let's go to the next slide, and it's going to be a whirlwind tour of data in terms of what we're talking about in terms of the data, of the types and variety of data, what it means and how it's collected, and really the amounts of data. Okay, next slide, please. So ultimately what we're talking about is we're taking what I would call unstructured information and figuring out ways how we can organize it, whether it's through machine learning, artificial intelligence, or us as human beings, figuring a way to organize it so we can actually identify and see trends quickly. Next slide, please. So just as a reminder, we have the way I think about data, it's really this one huge ecological system. So working from the bottom up, we're looking at a genomic or molecular level. We're looking at data that could be generated at the cellular or organ level. And for there are many of you that you may be looking at above, looking at the family or group affiliations, whether it's neighborhoods, community organizations, or looking at national and state phenomenon. And that we're looking at it also from a lifespan from pre-birth to older generations. Okay, so let's deep dive a little bit about what we mean in the next slide in terms of what we mean by data. It's a bit busy, but it's really to illustrate is that data means a lot of different things. It's real, it could be real-time, real-world data. That could be observational around what people look like or their diseases and phenotype. It could be genomic or metabolomic or microscopy. It could be location data, or it could be how people self-report their feelings or their actual behaviors. As we're getting into a lot more electronic data collection, we're looking at imaging, we're looking at electronic medical records. And what are we looking at? We're looking at exposures, whether they're chemical or particle exposures in the environment, but we're also looking at lived experience exposures, which could also include racism, discrimination, and prejudice. And then to the far right, we have to keep in mind that there are a lot of different ways now people are actually collecting this information and data, and a lot of it is through mobile sensors. Next slide, please. So as you can imagine, it means that there's a lot of data being collected. So if we think from the 1950s, there was a speculations that it took 50 years to double the amount of health knowledge out in sort of the universe at large. Today, I think it's even less than 70 days. I would say it's probably less than a month. So what we're really talking about is that the volume and the pace of data generation is really quite fast. The next slide, please. So think about it on a personal level. There has been speculation that each one of us actually are expected to generate over 1 million gigabytes of health data in our lifetime. So that's really the magnitude that we're talking about in terms of data. So next slide, please. So what this also means is how are we going to actually store this data, and how are we going to actually be able to utilize this data? So what you can see from this particular graph on the bottom particularly is really how technology has advanced the ability for us to store the data and actually use the data as at a decreasing financial cost. Next slide, please. And importantly, we have to think about veracity of the data. We need to make sure that it's trustworthy, that it's accurate, and we're able to define what that means, and that it's reliable. Next slide, please. And this is an adaptation of one of my favorite quotations from Toni Morrison, that we have to keep in mind that information and information uses and definitions, which we all are doing at some level, belong to the definers and not the defined. the one piece I just wanted to highlight in this particular slide is that we need to make sure, and it's really a cautionary tale, that we don't dehumanize people and our research participants into, quote, merely data points. Next slide, please. So I'm going to give you a high level of vantage point of what I'm talking about when I'm thinking about data science and what we're talking about resources. So the next slide, please. It's going to show you way back, not so long ago. This is until about 2014, and this is from a slide from Dr. Tony Karlovich at NCI. That around 2014, when we wanted to do secondary data use, we had a server. Some of us could have had it in our office where you downloaded the data. But because of this large amounts of data, at that time, only large institutions really had the ability to use the data. It was pretty expensive to store the data and to protect it for the security, around $2 billion a year. And it was really slow to download the data. For example, the Cancer Genome Atlas down at NCI, they had about 1 petabyte of data, and it would take almost a month to download the data. And this is within the environment where we were starting to see an increase of the rate of data generation. So what happened, next slide, is there was a spurt of, next slide, please, of how are we going to make this more efficient? And what we tried, and so the next move was really trying to what we would call co-locate these, quote, computational resources by making the access to these large data sets available without downloading the data. And then that way, researchers could bring their tools and pipelines to the data, and so they could combine their own data with this existing data. And this would be a workspace they could actually save and share and analyze the data with their collaborators. And the next slide, please. We're now in the new generation of this co-location, is what we're calling cloud computing, as really next pinnacle of democratizing data access and sharing, because it's cost effective, it's scalable, and it has computational capacity for a lot of these large data sets. So the next slide, please. So up to this point, I've been talking about the technology challenges, but I just want to highlight to you that it isn't only data and a technology challenge, that we need to keep in mind FAIR as the fair, accessible, interoperable, and reuse as the title of this webinar, but the human factors around caring. And this is the CARE principles about collective benefit, access, responsibility, and ethics that was generated from scholars in the tribal nations that we need to keep in mind. And next slide, please, is to also keep in mind the ethical challenges that really that include inclusion and respect. Okay, so we have to keep in mind that I've highlighted that the difficulties is that researchers have right now is that it's really hard to find and access the data. We have to figure out how to analyze large amounts of data and how to integrate it from a lot of different data points. So if you go to the next slide, so what you will see is as we start making these environments or data repositories more scalable and more usable by making the data harmonized so we can use it under a single sort of analysis platform for not only sharing and collaboration, and what I would call data analysis fluency. If you could click one more time, what you will find is that the exercise is actually becoming more social. Next slide. So what that social piece means that we need to focus on our communication. And it's within a backdrop of that we are now socialized to think that we can communicate clearly with merely 140 characters. So what this communication means, next slide, please, is that we are now in the world of multidisciplinary teams with diverse experience and resources. That it could include a biologist, a social, a psychological researcher who's identifying the research question or issues, trying to figure out the subsets, and really trying to figure out how can we explore the data. Click, please. And then the next piece is we need to have a computational scientist who's going to interact with the researcher on developing new tools and to how to share those tools to analyze that data. And next, click, please. Is also an algorithm developer who's going to help the team really test these programs and algorithms to ensure that we're actually analyzing the data in the right way. Next slide, please. So as what I'm proposing is we're actually in an age of innovation and revolution. And this is just to give you a time scale of what's been happening in terms of innovations. And so we can see from the turn of the 19th century, that is when there was a lot of transformations and revolutions around energy. And as we start looking into the 2000s, I think the new transformations are going to be around information. So the next slide, please. So what I'm propositing is that, in fact, we're in a data revolution because we're in a time and space where research is now a very data intensive enterprise. It's rapidly changing its scale, and that it is pervasive in our labs, in our clinics, and in our homes. Next slide, please.
This is a 3-minute warning.
Thank you. We means that we're changing the functional, what we're identifying is changing functional roles of genes across tissues. Next slide. Relationships across diseases. Next slide. Next click. And next click. And how these, I like this one because it's more in color, but it's showing that these diseases are all interrelated. And the next slide is that it's not only the diseases, next slide, please, it's also behaviors. And this is a study looking at the issues of weight and friendship networks. So the next slide is really talking about the data revolution of large amounts of data and really using it for secondary analysis as a way to identify trends and to improve research quality. So the next slide, please. What we're talking about is that science really isn't broken, although it may feel like that. Next slide, please. It's just really a lot harder. So next slide. So what we're talking about is a paradigm shift. Next click of going from next click is going to next click is a hypothesis way of thinking of things to more of a hypothesis generation. Next click, please. So NIH is doing this by having a strategic plan for data science to really try to organize the thinking and approaches to this data ecosystem. The last couple slides, next slide, please, is really the challenges that are before us. And it's the ELSI in terms of issues related to privacy and confidentiality, looking about how we're going to do informed consent, what is risk assessment and decision-making look like, and what are the benefits and harms. Next slide, please. We need to keep in mind that equity and disparity issues are at the forefront because data and information are not neutral and are prone to issues about exacerbating stigma, decreasing inclusion. And so we need to make sure that not only we're being inclusive, but we're also including issues around diversity and workforce. So next slide, please. So I challenge you to think of, if not designed to address equity in research and data, the data will perpetuate disparities and injustices. So next slide, what I'm challenging you to do is don't think outside the box. Think like there is no box. Thank you. That's all I have for you.
Thank you very much. Our next speaker is Dr. Harold Lehman. So Dr. Lehman is a professor of biomedical informatics and data science at Johns Hopkins University School of Medicine. And Dr. Lehman also serves as the informatics lead on the PATH PCORE-NET sites of Johns Hopkins, which he'll be mentioning. Thank you very much, and continue to drop questions in the Q&A.
Thank you. And building on what Dr. Odawang was telling you, I think I'll be focusing more on the reflexes that have to be unlearned and reshaped to investigators as they confront and live with these new types of data. Next slide, please. So we're talking a bit about how methodology changes, how reporting changes, some of the national efforts that you just heard mentioned, and what is, if you want open science, and we're talking about the Venn diagram really of open science, and then secondary use in the case that I'll be telling you of electronic health record data, what does a real computable environment mean to make all this happen? So next, please. Dr. Odawang already alluded to this, that we're used to thinking about science as hypothesis-based, that there's bespoke data collection, there's a pre-data collection analytic plan, you make the design to minimize biases, you have established rules of epistemology. Certainly those of you from, let's say, the FDA or other regulatory agencies, the rules are very important. So we know what is definitive. On the other hand, when you're talking about real world evidence, as you just heard, it's more about hypothesis generation. And what does hypothesis confirmation and validation really look like? We're not sure. The data collection from a scientific perspective is totally ad hoc, which means the data collection, the analytic plan has to be done after the data are collected. So the data collection actually maximizes biases, as I'll show. The rules are evolving, so we don't know what's definitive, and the world is changing as we speak, which is why your old reflexes have to be reevaluated. Next slide, please. When I sit down with investigators these days, I like to start off with a little directed acyclic graph. Some of them don't know what I'm talking about, and I have to do some education. Others are very familiar because they come from an epidemiological background. But if you look in the literature, even of real world evidence, there's very little of this going on, even though the data we have in secondary analysis is all full of confounders, collider biases, mediators. It's a morass of issues that do not get addressed. Next slide, please. As the saying goes, but wait, there's more, because the model on the right is what statistically minded people, epidemiological people are used to thinking about. But the real world evidence people are on the left. They get they get these data. Now, Dr. Odawang was talking about unstructured data. We can't even deal with that. We start off with the structured data like diagnoses, like numerical values for laboratory tests, procedure codes, and we have to figure out how to relate those data into the variables that the analyst wants. I'll explain what these little stacks of Lego blocks are in a minute, but the main idea here is the double arrows, that rather than the unidirectional, I've collected data and now I have my variables. Well, there's a site you have the data you need. Did your data definition, do you have the data that you want for the data definition that you specify? So there's the window of definition met by the data that you have. There's a lot of almost bargaining and compromise back and forth into going from the left to the right. Next slide, please. The type of constructs we have to build, and it's really is a building notion. We start off with let's say a code, or some general name for that as a concept ID. Then you have to figure out, well, if I have a code for sodium from plasma and sodium from serum, I have two different codes, but aren't those really the same thing? And the answer may be yes, and the answer may be no. Somebody has to make the judgment that for this research question, these codes are synonymous. Then you have a formula like a BMI, if we're still using that. And then you have to decide, well, what window of data, how long do I carry the data forward? In an adult, if I have a height from a year ago, that might be okay. If I'm doing pediatrics, a height from a year ago is not acceptable. So even the formula depends on the context. The transformation, what do I mean by on a medication? What do I mean by admitted to the hospital? What do I mean by length of stay? All those actually have to be defined in terms of some more primary blocks. When you get to a phenotype like experienced MI, you realize that that's both synonymous, but there's time involved, and there's windows, and there's washout windows, and finally a cohort definition is the quote Shakespeare local habitation and the time. When and where are you talking about? Next slide, please. The link at the bottom, which I think you'll have, is to a book that we put out for the National COVID Cohort Collaborative called A Guide to N3C. This is from the enclave analysis tools. The enclave is where we do the analysis. The chapter talks about the specific tools, but the workflow here that we've laid out is really generalizable. And it's interesting that it took about 2 years to figure out what the heck are we actually doing. What is the process of, quote, cleaning data, which really is about the process of getting from the raw data the variables you need for analysis. And even though most of us learn to do data cleaning in secret on our own, on the street, we're trying to figure out how to make it a more reproducible process. Next slide, please. Humorously, the whole analysis is left as oh, and by the way, now you do analysis, like magic happens here, and we're working out, well, what is the workflow analysis? If that's the workflow for data cleaning, what would we do for data analysis? And we're working on that as we speak. Next slide, please. There's a very nice article came out a couple of years ago. There are other checklists for methodology. You may be familiar with Consort for RCTs. There's Strobe for observational data, Record for EHR data. The one from Wang and colleagues is much more extensive. Basically, it's got about 150 elements that you need to pay attention to when doing real-world evidence. If you don't know what they're talking about, it becomes a great curriculum for what you need to know. And it should be something that you can instantiate for your research project. Next slide, please. Chen-Yu Lee and students and Karen Robinson from Hopkins and I did a review of the literature based on EHR data in the 2010s to look for the minimum required of assessed for missing data, methods that deal with bias and confounding, sensitivity analysis, and we only found, you can see about the level of any one of those is about 20%, and hasn't budged over 10 years. The proportion that did all three of those in the same study was only 3%, and hasn't budged in 10 years. Despite a number of federal guidances over those 10 years on how to do real-world evidence. So when I say we need to rethink our reflexes, I'm basing it on evidence like this. Next slide, please. So there are national efforts. Coronet is from the PCORI network. They're around 10 years old. They've have over 800 studies. Odyssey is more volunteer based and models itself as an open science collaborative. They have, quote, only 600 studies, pretty impressive for a mainly voluntary group. And the National COVID Collaborative, we've only been around for 3 years, so I think our 170 studies is a pretty good showing for those 3 years. Next slide, please. The methodological point I want to make comes from the Odyssey network. They were looking at hypertensive drugs. In the literature, they found 13 studies that provided 40 comparisons, and an arc or a chord of the circle shows a different comparison. I like to think, well, if each study was $1 million, then 13 million divided by 40 is about $300,000 per comparison. Next slide, please. When you have computers doing your analysis, you're able to do one study with 13,000 comparisons at the same time, which is about 3 orders of magnitude cheaper. So I would submit that going forward, anybody who funds research is going to say, why the heck are we spending $1 million or $300,000 when we can be spending 150? Now, before you all yell and scream at me, I'll point out that yes, there are reasons to do bespoke studies, especially when the drug is new. And at the same time, we don't know the rules of doing studies in a massively parallel environment like we're discussing. But that will be the future and is another reflex that we need to unlearn. Next slide, please. Finally, the communication. Dr. Ottawong kind of left implicit the how do you do communication in this new environment? And the truth of the matter is we still do it through articles. Somebody will publish an article, maybe they'll give you a URL to a database, but the evidence is all on paper. And evidence, PDFs are basically paper slapped onto a screen. They don't really help you understand the data any better, and they certainly don't use the power of the computer. So how do we get the computer, you put all the data into the computer, it generates the evidence, it then goes into a PDF, then some poor, you saw the slide of those other people using the brain imaging to do their own research, but those poor people had to re-implement a lot of details to do that secondary research. Why isn't the computer communicating directly to computer? How do you do that? And basically, where there's an effort called the EBM, Evidence-Based Medicine on Fire project, now called HEFCA, the Health Evidence Knowledge Accelerator, building on the FIRE resources that have been used, are being used to communicate patient data in an interoperable way. Now you want to communicate evidence in a machine computable way. And evidence is data plus method plus context. So you have to represent all those other things beyond just the data. Next slide, please. So this is my advertising. These are all the little sub-projects. I'm mostly involved in the lower one on the right-hand side, SEVCO, the Scientific Evidence Code System, where we're developing computer codes for methodology, which includes 75 codes for study design, 260 for biases. We're just developing the statistics and codes for statistical models so we can start communicating modern models and not just the old-fashioned t-test and even the regression coefficients. Next slide, please. And that concludes my proportion.
Thank you very much. So our next speaker is Dr. Anna van Gulik. Dr. van Gulik leads connections with government and funders at Figshare, where she represents Figshare to government partners. Dr. van Gulik takes the lead on everything from data curation and reporting, policies, and engagement, and is a strong advocate for open data and open science. And she'll be presenting a little bit more on Figshare and how the generalist repositories really help support that data reuse.
Great. Thank you so much for the introduction and the invitation to join you today. I'm going to take a little bit of a step back with this presentation to really the data sharing and how data sharing in repositories can greatly facilitate reuse of that data in the future through the FAIR principles. So next slide, please. So to start with just a little bit of context here, I want to make sure we're all on the same page about what a generalist repository is. So when you think about the NIH data repository landscape, there are many discipline or domain-specific repositories that exist, many of them funded by NIH or run by NIH institutes even. And these are should really be the first line that you go to for data sharing. That's what's recommended by the new data management and sharing policy, and different institutes and domains may have specific requirements that you use these repositories when they are available. And they're great because they provide very controlled metadata and offer a high chance of discoverability and reuse for that specific type of data. But as we've been hearing, the volume of data is growing. There's all different kinds of data, and there's a lot of data as well as other types of research outputs that do not have a domain or discipline-specific repository home. I should mention there's also institutional repositories which are sort of like generalist repositories, but are limited to those who are at a specific academic institution or organization. And so generalist repositories fill that gap for when data or another output does not have a home. So they offer the flexibility often to share any type of data, any type of file, and put it in a trusted repository. So Figshare is a generalist repository, and you may have heard of others, such as Dryad or Zenodo, or Open Science Framework. So next slide, please. So we have two public-facing repositories that Figshare runs. One is figshare.com, which was started 11 years ago and is available free of charge to researchers around the world to share any scholarly output. So it could be data sets, but it could also be code, posters, white papers, presentations, computing pipelines, anything that you can upload and share, you can get a DOI for. Also, about a year and a half ago, we launched Figshare Plus, which is a repository for larger data sets. Next slide, please. So with these, we strive to meet the NIH characteristics for desirable characteristics of data repositories as much as we can, and for fair data. So primarily, we offer flexibility. We try to meet researcher workflows with integrations of tools that are already used, using best practices such as persistent metadata like DOIs and ORCID integration, providing full open access so that everything that's published is fully publicly available and can be accessed free of charge. And in the case of Figshare, without even any authentication. So it is important to point out that most generalist repositories are for data that is not sensitive in nature. It needs to be, fully de-identified and can be made fully publicly available. And also offer a way to track the impact of that work, because as data sharing increases, especially in the context of NIH, it's important that we track what the impact is of that and how it's reused, such as through data citations. And then with Figshare Plus, we also help support data curation and metadata completeness during deposit for these larger data sets. Next slide, please. So I want to highlight Figshare's role in fair data, and this could really apply across any of the generalist repositories. So this is fair at Figshare, but I want to mention that it's together with a program called GRAY, which is the Generalist Repository Ecosystem Initiative. And this is a program out of the Office of Data Science Strategy at NIH. We're about a year and a half into it now, and this brings together 7 different generalist repositories, those ones that I mentioned. And so we're all actually working together to enhance our support for NIH data sharing and discovery and how we meet the FAIR principles. So here's kind of how generalist repositories fit into the FAIR principles when you look at the original paper presenting these. So I think you can't have the reusable without the findable, right? If no one can find your data, it cannot be reused. So as much as I understand this, this session of the webinar series is focused on the reusable, I will be addressing a little bit about the findable because they all go together really in my mind. And so repositories provide DOIs and metadata and discoverability, and that's the value of a trusted repository over sharing, on a private lab website or something like that, right? They're going to use structured metadata. It's going to be discoverable across search engines and indices. They're fully accessible, including through programmatic access, which is very important for reuse as well, providing interoperability. Again, maybe not necessarily at the discipline-specific level, but that standardized metadata, linking between outputs, flexibility, and then for reuse, having clear machine-readable licenses and metadata that gives context to the work. So when someone does find it and is able to download it, they can make sense of what the work is. Next slide, please. So here are these highlighted for the generalist repositories specifically. So beyond just data repositories, that flexibility is critical to share any research output and any file type. This applies at figshare.com completely, Also at Zenodo, at Dryad, they take only data sets. But this is a good way for maybe, you can put this certain data set, certain segment of your data sets in a discipline specific repository, but not every data output. Or maybe there's code that's needed to process the data as well, but that repository doesn't accept it. You can put it in a repository like Figshare and link them together. Discoverability and open access, open APIs, providing DOIs for all records is critical for discoverability and tracking of impact, so data can be cited, and use of other persistent identifiers such as ORCID for individual authors or ROR for research organizations and funding, as well as that standard structured metadata. And I'll show you some examples of that in just a moment. Next slide, please. So what are some data sharing practices that you could adopt that would help to make your data more reusable in the future? I've pulled out from the FAIR Principles paper a couple of things from the reusable section, and they focus both on metadata. So it states the metadata are richly described with a plurality of accurate and relevant attributes, and the metadata domain relevant community standards. So now you see my picture here of the road without the guardrails. This is sort of what generalist repositories are when it comes to this type of metadata, because working across all disciplines, we cannot be as precise in terms of our requirements for metadata. We have requirements for titles and authors and licenses and things like that that are standard, but this reusable metadata goes beyond that. And that's where actually we really need researchers to understand the best practices of their field and the best practices for data sharing generally, and take the initiative to add a README file and add a code book and put very descriptive metadata into a description section so that people can navigate this road of reusing their data. So you can use Figshare and generalist repositories to create FAIR records for all different research outputs of any type. So thinking throughout the research life cycle, what is any material that I'm using here? Maybe it's a stimulus set for a brain imaging study. What were the subjects looking at? Well, someone's going to need that to replicate your study or to combine studies. So it goes beyond just the data sets. Use DOIs and persistent identifiers for everything that you share. Link together those related materials. So feel free to mix and match your repositories and pick the most appropriate ones for each output, and then just link them via metadata and PIDs, and that will help everyone find them, even if they're not all in the same place. And I think that's something researchers can get nervous about, splitting things up, but that's actually a best practice. Provide that highly descriptive metadata that will support both discovery as well as reuse. So adhere to your own discipline-specific standards in documenting your data set. And because you can upload any file you would like and as many files as you'd like, there's plenty of flexibility to do that. And then, use a trusted repository to make sure that this is discoverable and accessible. Next slide, please. All right, so here are the examples of some of these. So here's a few examples of actually NIDDK funded research in Figshare. One is in Figshare Plus on the left, which is a larger data set supporting a publication. And so you'll see here that there's actually many files. And so you can upload hundreds of files or just a single file per what we call a Figshare item. And this has its own title, its own DOI, its own description, a license, and so forth. And the files that are uploaded here can be previewed. We preview over 1,200 different file types, so someone can review the files before downloading them. They can download all of the files or a subset. And on Figshare.com, you can upload data sets up to 20 gigabytes free of charge. Next slide, please. And here's a few more examples. So you'll see the previews that I was just talking about here. You can play this ultrasound video. You can see spinning molecules in action. And here's examples of, some images that have been uploaded. And images can be data, but they can also be hard to share. especially large ones. Multimedia files are important types of data. And that's actually how Figshare was founded. Our CEO and founder was a cell biologist, and he had a lot of videos of things moving from one side of a slide to another. And you can't put that in a journal article. And if you do, someone won't find it. So that's why Figshare was created. Next slide, please. And you can create records programmatically, which is a good way to make sure your metadata is complete without spending a very long time creating it over and over again. So if you have many things to upload, you can script actually the creation of items. You can upload files and create metadata through our Figshare API. And then these can all be accessed via the API as well for download, both the metadata and the files. Next slide, please. Integrations with other tools are again important for facilitating fair data because they make this more an automatic part of the research workflow. So integration with ORCID allows you to pull in metadata from your ORCID record for items. Integration with GitHub or GitLab or Bitbucket or your choice of version tracking software is important, and it's important to put snapshots of these into a trusted repository. Next slide, please. Giving every output a DOI is critical. This will provide persistence. There won't be link rot. These will always point to this record. If it's removed, it will be tombstoned. Gives you a clear reference point to in articles, even on web pages. If you do want to link to materials on a web page, use the DOI to a repository. And with this, also the metadata is then stored with the DOI provider, and that gives that indexing and discoverability. Next slide, please.
Anna, this is the 2-minute warning.
Okay, thanks. I think I'm tight on time. Here's an example of giving a DOI to a GitHub repository so that it's citable. Next slide, please. And you can also create collections of items where you have a DOI for many different public outputs, and you can group them together with a single DOI. Next slide, please. These DOIs can be reserved in advance, and they're also version controlled, which is an important feature for planning data sharing. Next slide, please. Here's the structured metadata for all outputs. So this is our edit item page on Figshare so that you can provide that context and discoverability, use PIDs, link to NIH funding. Next slide, please. And here's what it looks like published on the formal page. And then on the next slide, please, you can see the JSON. So here's on the back end, or if you accessed it via the API, how you would find that structured metadata. And this is shared with DataCite, which is an international collaboration, which is what mints DOIs for us and holds this information. Next slide, please. And it's important to use these persistent identifiers, not just for that item you're sharing, but also for linking to related materials. So we have a section there where you can choose from multiple different persistent identifiers to link to, for example, the related publication for a data set. Next slide, please. And here on the back end is what that would look like, including ORCID identifiers for each author and RORs for organizations coming soon. Next slide, please. And it's really important, especially for NIH, that you link to your funding resources. So we do have an integration with our sister company Dimensions, which is an index of funding and scholarly outputs. So you can find your specific NIDDK award, and link to it through the award ID, and that will link to Dimensions, the Dimensions record about that funding source. Next slide, please. A license is critical for reuse. On Figshare.com, we offer CC BY, CC0 licenses and some software licenses, and please choose the most open one possible for your data sets, but these will be machine readable. Next slide, please. I think I have just a couple more. And so then accessing the files and metadata can also be done via the API. So this is also very important for someone who wants to reuse data, especially large data or many data sets. Next slide, please. And so again, here's how you would do that with the Figshare API. Next slide, please. Here's an example of actually a data visualization tool that someone built that's continuously updated through API pulls of this data set found on Figshare.com. Next slide, please. And here is when you look, when searching on the API for that item, next click through, and then you'll find the JSON on the back end there for it. Next slide. And my last slide is just the discoverability, which is so key for these FAIR outputs, to offer opportunities for innovation and collaboration. These outputs, when they're well described, have a much better chance of being both found and reused. And so then someone searching through any one of these indices listed here might be able to find that work, just searching by topic or method, and be able to find all of the outputs that are needed, whether they're in one repository or multiple, that are needed for replication or reuse. And next slide. That's the end of my presentation. The next two slides just have some links to guides for NIH materials and for getting started using Figshare. Thanks very much.
Thank you very much to all the speakers, and we're going to turn it over to a quick discussion moderated by Dr. Odewang. Again, I'm going to drop some more information into the meeting chat, but continue to put questions on the Q&A.
So first of all, thank you all of you for spending time with us. And at some level, you got a whirlwind tour of reuse. And so I think some of the main points is this is a train that is out and moving quickly. And I would hope that you experience this as being a very exciting time where the science is changing, not only in terms of the data that is available that you can use, but it's really a way to expand the type of scientists that you're working with, to really be able to address and answer much more complicated questions. I think we've answered all of the questions that were posed in the Q&A. Let me just make sure. So I guess I just want to open it up to the other two speakers. If there are any last words you would like to impart to the audience. Dr. Lehman? I think you need to turn on your, there you go.
No, I just said I'm thinking about how to integrate everything that's been said. But I think, I guess I would ask to you, do you distinguish between secondary use of, how do you distinguish between data that was collected for a specific research purpose and its reuse versus the real world evidence type of thing that I alluded to and half your slides alluded to? And how do you distinguish those, or do you distinguish those two?
Okay, in less than 30 seconds, I will give you a not very satisfying answer. I actually will say it's a bigger bucket that I don't distinguish it. And I think it's an important distinction that you're bringing up because I think it's very different. I tend to focus on how people are using the data, and I always go back to the informed consents. So part of it is however they're consented, whether it's real world data or purposeful data for a particular question. It really is important that all researchers respect the conditions about what the research participants agree to. And I think research participants may not always understand the nuances between real world data and what you're talking about. Dr. Van Gulik? Any last words? I think we're at time, but I will give you the last word.
Time. Just share your data in all the repositories, and there's lots of resources at the repositories at your institution and at NIH. So seek out those resources, and this is an example of one of them at NIDDK.
Thank you, everybody, and thanks again to all the folks who attended and our speakers. I just want to do a couple quick reminders. There will be a survey that pops up as you exit, or I've dropped a link into the chat. Materials are all going to be available on the meeting registration site, and there will be an email that goes out with links to that. So thanks, everybody, and please keep sharing your data.